the article that came out showing that a dead salmon in an fMRI showed brain activity changed the way you conduct fMRI research? This is a very precise question. This is a really good question. Yes, 100%. So I don't think any student in neuroscience that does clinical imaging can go through their career and not get exposed to this article. It's very commonly journal clubbed. It's hammered into every student very rigorously that this multiple comparison problem is a big deal. So what people who aren't in neuroimaging, what this person's referencing is in fMRI, the classic approach was to essentially do hundreds of thousands of t-tests to determine neural activity. So let me back up. Sorry, 1 second. So a normal brain image, if you take a whole brain image, can have hundreds of thousands, even millions of voxels, and these are just three-dimensional cubes. And the origin of brain imaging was to do what's called statistical parametric mapping. And so it's a method that takes advantage of the nature of neuroimaging data being high in spatial resolution. So you have hundreds of thousands of millions of spatial data points. And what the statistical parametric mapping would do, essentially would do a t-test at every single voxel in condition A, like moving your knee, versus condition B, which would be at rest. And it would ask the question, is the bold signal in this voxel, the bold signal is the blood oxygen level dependent signal. And so that is our surrogate for neural activity. So it asks the question, is the activity, that blood signal in this voxel higher in condition mu versus condition rest? And is that probability above a set amount, mostly using the typical like p.05 that we all are familiar with? Now that would normally work fine if you just had a few voxel comparisons, but the problem with that was in a brain imaging data set, you could have hundreds of thousands of comparisons. And so a p.05 threshold means 5% of the time you will have a false result. And so what that meant is you could have hundreds or thousands of false positive results with brain imaging. And to illustrate the point, Eklund's group, they put a dead salmon in an fMRI machine, and I think it won the Ig Nobel Prize, which is like a joke Nobel Prize for a bit of a tongue-in-cheek experiment or article. But it was good because it kind of really drew attention to this problem in the field. And the problem was you have to correct for all those hundreds of thousands or millions of comparisons. And what they showed was that a dead salmon had a little bit of brain activity because they had false positives when they put them in the scanner because of the many, many comparisons. And so the modern software, and for me, even by the time I was trained in neuroscience 5 to 6, when I started almost a decade ago now, you can't have a paper published with that sort of flaw. And so none of our lab's papers and none of the papers that related to orthopedics or ACL, nothing in the last 5 to 10 years, really, not many of them have it, maybe some closer to the 10-year mark, but there's really not many papers that exist today that survive, that can get through peer review with that sort of flaw. And so how I do neuroimaging today is we use a lot of multiple comparison correction techniques, and a lot of them take advantage of the nature of fMRI data being spatially correlated, in that an individual voxel will very rarely be active and all the ones around it not active. So this is called, it uses Gaussian random field cluster-based inferencing, which is a big term, which basically means it checks that one voxel if it's active and all the voxels around it. And if a lot of them also are active, then it increases the probability that that's a real finding because you have multiple voxels in a brain region that could be active, versus just a single voxel in the middle of nowhere. That's more likely to be artifact or to not be real or be a false positive result. So the dead salmon paper, I think, changed the entire field, and it changed it for the better. And it definitely influences how I run statistics. And we don't report uncorrected data anymore for that risk of false positives. So that was a cool question.